You can see for Huntington's disease, basically the brain is the most affected area. Usually the brain just either doesn't function for younger people, or in adults begins to actually lose function, and it's also lose its structures. You can see here on the left a brain that was affected by Huntington's disease versus a normal brain on the right. Sorry, I didn't want to put this on the last one and risk of running out of time, because having to edit these and smash them into 15 minutes to get them onto YouTube can be a pain in the butt. So moving on to the next subject, karyotypes. And basically karyotypes are just a picture of chromosomes, literally a picture of chromosomes that they can take, and you can determine quite a few things from that. And the easiest thing to see on here is if there is an incorrect chromosome number. So you can see here both of these karyotypes here represent individuals with the correct amount of chromosomes, 2 for each number, 1 through 22, and then also 2 sex chromosomes, an X and a Y here, or Y and an X I guess in order, and 2 X's here. So we all know which one's male and female, right? So like you saw earlier, a person with Down syndrome would have 3 chromosome 21s. You could have certain disorders involving the sex chromosomes, which typically involves fertility problems. But some of those disorders could be just sections of chromosome missing, like Turner syndrome, which is an XO. It could be for like this female here. If she had Turner syndrome, it would be either missing this X chromosome, or it could be just a large part of this X chromosome was missing. So different ways to, I guess, analyze these, but the most important is to determine what's called aneuploidy, which is an incorrect chromosome number. Polyploidy, which would be more than the usual amount, 47, very rarely 48, but 47 can happen. That's common in the ones you've seen on the previous notes. The next section we're going to talk about is non-Mendelian inheritance, and basically this would, if Mendel studied this, he probably would have went nuts and would not have figured out nearly what he did figure out with his pea plants, which showed simple dominant and recessive traits like tall and short. In these cases, there is no, or there are very few things that kind of follow Mendel's rules, Mendel's laws. The first one is incomplete dominance. To give you an example what this would be like for Mendel's pea plants, this would have been like taking a short plant and a tall plant, and instead of getting all tall plants in the first offspring generation, the F1s, incomplete dominance would create medium-sized plants. So again, this would have been really tough for Mendel to figure out. Codominance would have basically applied to something like the colors of Mendel's pea plants, say the purple flower versus the white flower. Codominance would suggest that a white plant crossed with a purple plant would create offspring that were both purple and white. That's what codominance is. It's seeing both at the same time. And then sex-linked is obviously something that Mendel could not have described because he didn't know much about sex chromosomes or chromosomes at all. And you'll see sex-linked when we talk a little bit more about doing the Punnett Squares. Like for instance, red-green colorblindness is a sex-linked trait, and it's actually an X-linked trait, X-linked recessive, meaning you have to have, for girls, 2 X's that are affected with this mutation, which is very, very rare. For males, it only takes 1 X, because that's all we have is 1 X. Therefore, that's why you saw it was 8% in males and almost non-existent in females for red-green colorblindness. And we will do some of these practice problems in class on the back of your notes. Last thing we'll talk about are chromosomal mutations. A lot of the disorders we just learned about were actually gene mutations. However, we're going to cover gene mutations in the next unit and just cover chromosomal mutations right now because they're a little bit easier, and once you see these, I think the gene mutations will make a lot more sense. And for now, if you just want to save yourself some time, these 4 chromosomes here on the left are kind of the starter chromosomes. If you want to save yourself some time, just label each one of these little boxes and each of these 4 chromosomes A, B, C, D, E, F, G, H. For all 4 of these, then just put an arrow, and this is going to we'll do this in class, but you'll see how the chromosomes change depending on what type of chromosomal mutation that takes place. So again, for all 4 of these, just label them A, B, C, D, E, F, G, H. And then these are going to have some kind of mutation to them.